Hey guys, this is Mr. Mahmood, and you're watching the third and final lecture for the biochemistry unit. Now, I know this unit has been pretty long, and there's been a whole lot of detail to deal with in it, but it's critical that you guys are really comfortable with all the information you've learned in this unit because we really are laying the foundation for the entire school year. We're going to be referring back to organic compounds multiple times over the course of the year because of how critical a role all of these different categories of organic compounds play in all living things and all living functions. So we're finishing our discussion here with the fourth and final category of organic compounds, which are proteins. So we've talked already about carbohydrates, lipids, and nucleic acids. We talked about the general characteristics of monomers compared to polymers. Now we're really going to focus in on proteins, and then we're going to spend a whole lot more time on proteins a little bit later, but it's really important you get that base now so that when we come back to it, it'll make a whole lot more sense to you. So we'll talk about proteins in detail, kind of the basic structure and components, major functions of proteins, and then we'll be able to talk about a particular function of proteins and a category of proteins that we call enzymes. And enzymes are going to be very important. You're going to spend some time in this unit on it in your classes, but then you're going to come back to enzymes multiple times throughout the year. So we'll focus particularly on enzymes and their functions within the cell. And then we're going to finish out kind of with a basic discussion of what came first. We've already kind of been going over the monomer and polymer relationship, how one is built from the other, but now we're going to talk a little bit about where we can figure out how these organic compounds came about, and some theories out there, suggestions of how those first organic compounds could have been made, which of the four categories they might have been, and how they could have led to the formation of the rest. So let's get going on this. It's, again, the last video of the unit, so you can breathe easy for a little while once you finish out with this one. Okay, so proteins, just like the other three organic compounds, have what's considered a monomer, and then the polymer would be many monomers put together. Remember, for carbohydrates, the monomer was the monosaccharide, or the simple sugar, and then the carbohydrate, the complex carbohydrate, was a polysaccharide or a starch. For a lipid, it was a little more gray. It wasn't so much a monomer compared to the polymer, but more of the monomer being the parts of the lipid, like the glycerol backbone and the three fatty acid tail extensions were considered the monomers, and then the polymer would be that whole triglyceride together. For nucleic acids, we briefly went over that. They spent a whole lot more time later when we get into DNA and RNA, but the nucleic acids monomer is the nucleotide, and then the whole chain of nucleotides combine to make your nucleic acids. For a protein, we have something that's a single piece by itself, just like a carbohydrate, just like a nucleic acid. This single piece is what's considered an amino acid. Okay? Now, what you see here is the basic structure of an amino acid. Now, there are 20 different forms of amino acids, and I'll never expect you to memorize all the variations of the different forms. You might get to that a little bit later if you continue on in biology, further on in AP or into your college careers. But for now, what I want you guys to understand is you can pretty much represent all 20 of the different amino acids with this basic structure. And the one thing that's different about all 20 of the amino acids is what you see extending down from that first C, that C that's right there in the middle. It's called an R group. It's basically just a variable, kind of like in algebra, you represent X to mean a lot of different things. This R represents a lot of possible extensions that come off of that carbon. In fact, there are 20 possible extensions that come off of it, and that's what makes every single amino acid different. So that's critically important when we start talking about how things come together through dehydration synthesis and break apart. It doesn't matter which amino acid we're talking about, they're all going to combine and separate the same way because the basic structure across the top there is identical. Now let's start talking about how you go through the combination of these monomers, again, the amino acids, to form the polymer. In this case, a polymer is going to be multiple amino acids put together. And because the structure is identical in all 20 with the exception of that R, we really just need to focus on this one basic example, and you'll understand it happens like this every single time. These single pieces will combine to make chains. This is very similar to carbohydrates. Remember, carbohydrates, the monosaccharides, just formed linear chains, like long chains connected to each other. That's exactly what's happening here for the most part, at least at the beginning, when we talk about the formation of these long chains of proteins. It happens here when you combine the basic carbon with a double bonded oxygen and an OH there on the left of the first amino acid with the amine group, which is a nitrogen with two hydrogens from the left side of the right amino acid. So when the two combine together, they're going to form the same basic components and processes that we've talked about before with dehydration synthesis. Dehydration means you're going to lose the water, and synthesis means you're going to combine. So the same process is going to happen here. But if you remember, we talked about this with carbohydrates and with lipids particularly. When these two components combine to start making your polymer, there's always that oxygen that was sort of the connector. You guys remember you had an OH on either side, so 2 hydrogens, 2 oxygens total. Of the 4 elements, 3 of them combined to make water, and you were just left with that one single oxygen that was the connector that made this turn into a chain. You remember that because there was an OH on either side. If you'll notice, looking at these amino acids when they combine, we're going to focus everything in on these elements here at the bottom. You'll notice you have exactly the right elements for water. You don't have an extra oxygen here because this amine group on this side for the amino acid on the right, there's no O down here. It's simply an H. So when all of these elements combine, that'll be your water. So all 3 of those elements will get eliminated, which means there's no connector anymore. So when we talk about a bond holding these together, it's no longer going to be a bond that's just sort of extending out from underneath. There's going to have to be a bond directly in the middle of the chain here. Again, you're going to lose a water molecule in the process, the dehydration, and then the synthesis is going to occur to allow them both to bond. And that single bond that combines that carbon and that nitrogen right there in the middle is what we call a specific type of covalent bond known as a peptide bond. So you're going to hear the word peptide used multiple times in relations to proteins, and all that represents is the bond that's holding these amino acids together. So once I've taken these 2 amino acids and combined them together with forming a peptide bond, losing water and combining in the process, this is that process of dehydration synthesis, forming a single peptide bond between 2 amino acids. And this process will continue to happen one amino acid to the next and just continue to add from one to the next until you get this long chain of amino acids. And that full chain of amino acids is what's referred to as a polypeptide. Poly means many. Peptide just means the bond. So a polypeptide simply means a chain that has a bunch of peptide bonds. So the monomer of a protein is the amino acid. The polymer officially is called a polypeptide. Many peptide bonds holding together this amino acid. Let's talk about the major functions of proteins. You've probably heard before if you're in athletics, like a football player or anything like that, where you're trying to build as much muscle as possible, they'll usually encourage you to take in a lot of protein in your diet, maybe take protein shakes or whey protein powder, things like that. That's because your physical fiber structure and a lot of your structural components of your body are high in protein. So if you're trying to build more of this structure, you're going to need to take in more proteins, right? Proteins play an important role in the structure of your body. It's one of the major components. And a unique fact here when we talk about nutrition, because we've kind of already been getting into nutrition with carbohydrates and lipids and things like that, proteins are essential for your nutrition because of the 20 amino acids that we've talked about before, only 12 of them are made naturally in your body. They can be recycled from other protein components through hydrolysis, kind of breaking apart the proteins and using the amino acids over again. But of the 20, only 12 of them can actually be reproduced and recreated by the body. The remaining 8 are what we call your essential amino acids. These are the amino acids that you cannot get naturally. You have to bring in through your diet. So this is why they always say it's very important to have a certain amount of protein in your diet, because you have to be able to get those 8 amino acids in. Think about it. If you didn't have the 8 amino acids, you'd be making proteins out of just 12 of the 20 amino acids. That means any protein you're making can only be made of those 12 amino acids. And we're talking about proteins that can be hundreds of amino acids long and all unique variations. It's like saying you have 26 letters in the English alphabet, but you can't use the first 8. If I told you that, do you think you'd be able to have a normal conversation for the rest of the day? Absolutely not. Imagine that. It'd be pretty crazy, right? It's the same thing. If I told you to make proteins with only 12 of the 20 amino acids, most proteins wouldn't be made. So you have to make sure you're getting your essential proteins from your diet so you have all 20 available in your cells to make all the proteins that you need. And then specifically we're talking about the physical components, the physical structure of the body, particularly your muscle fibers, and then exterior components like your hair, your skin, your nails. All of those things are very high in protein content. So those cells in particular need even more proteins available to them to stay structurally important. So on a large scale, proteins play a major role in the physical structure of the body. But proteins also play a critical role when you start talking about your endocrine system, which is responsible for making hormones. We're going to get into that later throughout the year as well. A lot of the hormones that your body makes are actually proteins. They're actually made of proteins, and they're called peptide hormones. So why do you think they're called peptide hormones? Yeah, exactly. They have peptide bonds. Crazy. It's a crazy concept, right? That scientists would actually make sense in naming things. But in this case, it's true. These are called peptide hormones because they're made of proteins. When a protein is made, the whole polypeptide, they fold over themselves and start making these unique shapes. They make unique shapes on the surface as well. Because of the way they folded, their surface have all these different ridges and grooves and things like that, and each one of those is going to become very, very important. So this is kind of the realistic drawing. Here's more of like an animation, right? So imagine that each one of these gray things represents a uniquely shaped protein. Because of the way it folded, it created some unique shapes on the outside. So these unique shapes on the outside will serve as sort of like a dock for certain chemical reactions to land in. And if a chemical reaction can land in it, the components of that chemical reaction can land in that groove with a perfect fit, then that reaction will actually be able to happen faster. And these are the types of proteins that we call enzymes. So we're going to talk about enzymes a lot because it's an enzyme that actually gives certain chemical reactions. Each enzyme is unique because it has to fit perfectly with the components of the reaction. It's the enzyme that can give certain chemical reactions the ability to happen much more easily for a very specific reason. So enzymes are actually able to sort of speed up and make chemical reactions more efficient. And without enzymes, chemical reactions would take way too long to happen and no living things would actually be able to survive. Enzymes are so critical in just about every chemical reaction that happens in all living things that if we didn't have them, then it would take way too long, it would take way too much energy for chemical reactions to happen and no life could exist. So these enzymes are very important. So let's talk a little bit more about this concept of how enzymes affect chemical reactions. So what I have here is a basic graph. On the y-axis is energy and the x-axis here is representing time, right? It's not just thinking about the positive components because as we're going to see it's going to kind of flow from positive to negative, but we're going to start here at the 0 point. With any given chemical reaction, I hope you guys have kind of remembered this all from middle school. With any given chemical reactions you have what's called the reactants and the products. The reactants are what you start with in a chemical reaction. They're basically the materials. And then the products are what you end with. I make the reference like if I'm making a cake, right? My reactants are my ingredients, like the eggs, the water, the sugar, all the other stuff, right? If I'm making the products, the product would be the actual cake. When it's all said and done, all the reactions have happened, everything, the mixing has occurred, and my end result is what we consider the products. So if I refer to any given chemical reaction, the reactants are what you start with, the products are what we end with. Now let's give a specific example of a chemical reaction that you're going to deal with in this unit and kind of connected to this with a specific lab. And that reaction is working with a chemical called hydrogen peroxide. Hydrogen peroxide is H2O2. If you know anything about it, hydrogen peroxide is something you probably have in your medical cabinet. For cuts, for cleaning cuts and wounds. You put it over a cut, it's usually in a brown bottle, you pour it over a cut and it kind of disinfects your cut for you, right? And you'll learn a little more about what exactly is going on with hydrogen peroxide in your classroom, but just the basic component here, that's what hydrogen peroxide is, right? In reactions, when hydrogen peroxide actually goes through its reaction, it turns into two basic products. You should know when you pour hydrogen peroxide over a cut, you start seeing a whole lot of bubbles. So you should know that one of the products is a gas. And in reality, looking at the components, we usually deal with only two gases in biology. It's either carbon dioxide or oxygen, right? Of the elements that we're working with here, we have hydrogens and oxygens to work with. What gas do you think can be produced from these? Would it be carbon dioxide or oxygen? Oxygen, right? Yeah, there's no C's, right? So those C's don't just come out of nowhere. So I'm making something with these ingredients, which means my products have to have these ingredients in them. So the only gas that I can create from it is actually oxygen gas, or O2. So the other product is actually something that's completely harmless, which is what you can make with the remainder of this stuff, which is H2O. And you're going to talk about balancing chemical reactions and things like that later. But basically, if you wanted to balance this out, put a 2 in front of the hydrogen peroxide and a 2 in front of the water, Bob's your uncle, everything's balanced out. So this is the basic chemical reaction you guys are going to be working with. Hydrogen peroxide by itself is actually fairly toxic for your cells. Your cells can't really function and work well when hydrogen peroxide is present. So it's necessary that hydrogen peroxide actually goes through this reaction to turn it into basically harmless components. So the reaction itself, if we represent this graphically, we'll say our starting point, we'll put, say, right here at our balance line, our baseline of energy. So this is our hydrogen peroxide. This is our H2O2. If I want the hydrogen peroxide to actually go through the chemical reaction, I have to kind of give it a kickstart first. I have to jump start it. So it's sort of like a car. You have to put some energy in to get the things, all the gears moving to get the car started, right? Another example that we make reference to all the time is a roller coaster. Think about the older model roller coasters, not necessarily the newer ones, but if you've been to Six Flags or anything like that, like the big wooden roller coasters, they all have one thing in common. In terms of the engineering and the structure, they all have one thing in common, and that's that big hill right at the beginning of the ride. Every single one of those has that big hill right at the beginning, and then all the other ones kind of are smaller in comparison. There's a reason for that. That first big hill gives all of the energy needed, that kinetic energy or the momentum for the car to be able to work all the way through the rest of the ride, right? So it's the same thing for a chemical reaction. If I actually want this chemical reaction to take place, I first have to give it that kickstart. I have to put a whole lot of energy in to get things moving so that the chemical reaction can actually take place, and this is sort of like the roller coaster. And what I end with are my products, or what are sort of my end result. So let's say I have two components of products here at the end. So the two products in this case are oxygen and water, just like we see in the chemical reaction above. So this amount of energy that we need to start any given chemical reaction, if I were to just sort of think vertically from here to here, the amount of energy needed to start any given chemical reaction is called the activation energy, also written as EA, if you ever see that. EA is also known as the activation energy. It's the amount of energy you need to start a chemical reaction. It's that first hill, or how do you get things kick-started, right? For any given chemical reaction, that activation energy is relatively high compared to basically the amount of energy available in the cells and things like that. It takes a lot of energy to get this thing kick-started, right? So this activation energy is what's needed to get the reaction moving. And then once it gets moving, then we can talk about the components. And in this case, we're taking something larger, breaking bonds in order to produce smaller components. And you guys remember, we've talked about this already as to why lipids are such a good storage of energy, because they have so many bonds, right? We've talked about how the bonds of a chemical reaction are where all of that energy is stored. So if I'm trying to take something and break it into smaller pieces, I have to break bonds in the process. And when I break a bond, I release all of the energy inside of it. So that's why our products down here are actually a little bit lower than where we started, because by breaking the bonds and releasing that energy, what I'm left with has a little bit less energy than what I started with. Okay? Conversely, if I were to take a chemical reaction that I was starting with smaller parts and actually making a bigger piece, that means I have to actually put energy in to form new bonds, which would make this curve look something like this instead. So if I were taking smaller pieces here and then combining to make one bigger piece here, I'd have to put energy in order to form a new bond, which means the energy level at the end is actually higher. What I end with has more energy within it than what I started with, because I'm adding bonds here, but I'm breaking bonds down here. Because I broke bonds, I released the energy to make one big thing into two smaller things as my products, right? So this is the basic graphical representation of a chemical reaction. This is the normal components and using the full activation energy necessary for that reaction. Now, the reactants, what we start with in a chemical reaction, actually have another name. They're also known as the substrate. So if you ever hear me refer to a substrate, I'm referring to the reactants, the beginning of a reaction, whatever that might be. The parts needed at the beginning. Usually maybe it's just one part needed at the beginning or two parts needed, something like that. So my reactants are my substrate, or my starting point. My products are my end point. In this case, it needs a lot of energy to get things moving. Now the problem here is that the amount of energy needed for this reaction is actually way too high for the cell. It would take way too long to build up that energy, and then by the time you had all that energy, you now would be depleted and you don't have enough energy for the rest of the cell. So the relation I make, the analogy I make here is, think about, say you wanted something that was really, really expensive. Let's say I'm really into shoes, okay, and I really want this new pair of shoes. They're $300, right? But I really want them. But I only have 150, right? So I have a few options, legal options. I'm not gonna talk about the other options out there. But basically, I can do one of two things. I could either wait, continue to save my money until I finally saved up $300 and I can go and just buy it for full value. Or I can start shopping around, I can start looking online, maybe Amazon, eBay, things like that, and try to find it where it's not so expensive. Maybe I can find it on sale. And if I can find it on sale, I already have the energy I need. I don't have to wait for myself to save up anymore. I can go get it right away. So I have those two options. I can either just wait, keep building up, keep saving the money until I finally have what I need, or I can find it on sale and go ahead and get it right away. I don't have to wait anymore. And that's basically what an enzyme is actually able to do for these chemical reactions. So an enzyme effectively is going to allow reactions to happen with less energy. So basically, if I had an enzyme present for this chemical reaction, it wouldn't take all of this energy to get things moving. Instead, it would take significantly less. Let's say I only needed to get up to about this mark. So now, this chemical reaction, even though you're starting at the same point and you're ending at the same point, the path that got you there is different. So the purple line that you see here is the same chemical reaction, but this time in the presence of the enzyme that allows this reaction to happen faster. By lowering the activation energy, I allow this chemical reaction to happen faster because it doesn't take that long to build up that energy because there's not as much energy needed. And that's great for your cells because one, the reaction happens quickly, and two, you have extra energy. You have energy available for other things now because you're not using so much on this one thing. Just like my shoe scenario, I've already gotten my shoes with $150, so the remaining money that I save later, I can use on something else. I don't have to just put it all towards one thing. Just like your cells, they want to be able to work efficiently and use the least amount of energy for any given chemical reaction. So enzymes are beautiful for lowering activation energy and speeding up reactions. But again, like I mentioned, you can't just have a one-size-fits-all kind of enzyme. It's all based on those unique grooves and shapes that allow for chemical reactions to take place. So when we think about the basic structure here, remember this picture before, it's these unique grooves that are on the enzyme that are actually going to allow only certain substrates or only certain reactants to come in. So this gray figure here represents the enzyme. And in this case, this green figure represents the substrate or the reactants. In this case, it looks like we have one full structure, one solid structure here because it's all the same color, and it has this really unique shape to it. So if I want to find an enzyme that is going to allow me as this substrate to break down faster, I need to make sure that my enzyme has the exact same sort of groove shapes on its surface, or I should say the exact opposite groove shapes on its surface so that I can fit with the other like a lock and key, right? So this unique area on an enzyme that has really unique groove shapes is called the active site. So this is the active site of this particular enzyme. And if the substrate can fit the active site of the enzyme, then they combine together, which lowers the activation energy in this region, so this chemical reaction, this one piece can break into two and become our two products significantly faster. If this had to happen out on the surface without any sort of an enzyme present, this whole reaction would take a whole lot longer and probably wouldn't happen. So there would be problems in the cell because this would take way too long and require way too much energy. But if it can fit into that active site, then the energy level will be lower, or the chemical reaction can happen faster. So that's what this enzyme does. Enzymes lower the activation energy to allow chemical reactions to happen faster, but only if the substrate can fit perfectly into the active site of that enzyme. Okay? And again, enzymes are very specific for certain reactions. You have thousands of different enzymes in your body. Each one of them is critical for the overall functioning of your cells. Very, very important that you understand why enzymes are so important, and we're going to talk about all different types of enzymes over the course of the year. All right, so we have a basic understanding of what proteins are, particularly how enzymes work and what role they play. You should also know that enzymes are very specific to their environments. So because they become very well adapted for certain reactions, they've also become very well adapted to certain environments where the reactions commonly take place. If you change the environment, some certain components about the environment, the enzyme won't work as well. Okay, so there are three basic categories of environmental factors that can affect how well an enzyme works. One is the temperature of the environment. Let's say that you're working with an enzyme that's really good within the human body, which is right at 37 degrees Celsius or about 98 to 99 degrees Fahrenheit. If I took that enzyme and tried using it in room temperature, which is about 25 degrees Celsius or about 72 to 75 degrees Fahrenheit, it wouldn't work as well because that's significantly cooler than it's used to being in. Right? And vice versa, if it was an enzyme that works well in room temperature and you try to use it in the human body, it wouldn't work very well also. So by changing the temperature, it can drastically affect how well the enzyme is able to work. Usually it'll make the reaction take longer if you pull away from its normal range. If you keep it in the normal range, it speeds up reactions because it really works well to lower the activation energy. pH is another good example. If I have something that works really well at a neutral pH, which is about a 7, I couldn't use it in a really acidic environment, which is anything lower than 7, or a really basic environment, which is anything higher than 7. Again, because by pulling away from its natural environment, it won't work as well to lower activation energy, which means the reaction happens significantly slower. And another component that we usually talk about deals with the concentration of enzymes. The more enzymes there are available on the surface, and particularly the more active sites are exposed for substrates to come in and attach, the faster the chemical reaction can happen. That's a basic analogy of going to a grocery store. If there are 10 people, each one with a full cart, all going to the front of the grocery store at the same time, and there's only one register open, it's going to take a while because that one register has to sit there and process all 10 of the people. But if you have 10 people coming in, but there are 10 registers open, then all of us are going to get through the line much faster, since each one of the registers can take us at the same time. Multiple customers can be checked out at the same time. Substrates and enzymes work the same way. If you have multiple exposed enzymes and multiple exposed active sites available, then the substrates don't have to wait in line for their normal active site. They can all just go wherever they fit, and multiple reactions can be happening at once. So the higher the concentration of enzymes, or the higher the exposed active sites, then the faster the reaction typically works. So pH, temperature, concentration, all three of those have major roles in how well enzymes work. You guys are going to be doing a lab kind of practicing that and testing it out. All right, so the very last part of this unit, and what's going to pull us into the next unit and start talking a little bit about a discussion of, where did all this come from? So what now? We've got this basic understanding of why all of these things are so important in the body, but where do they come from in the first place? We're going to start talking about theories related to early Earth and the early development of life and of organic compounds and of everything kind of branching off from that. In terms of these four categories of organic compounds, the basic premise and the basic idea is we want to figure out how it could be possible to make organic compounds out of inorganic components. Because we know at some point in early Earth, there had to be formation of basic organic compounds, because without these organic compounds, like we've talked about, you can't have any kind of life at all. This is a question that was highly debated in terms of all these different ideas and concepts and thoughts and principles, but the first group of scientists that were actually able to prove that you could make organic compounds from inorganic compounds were a combination of two scientists named Miller and Urey. Basically what they did is they tried to simulate early Earth's atmosphere by putting a bunch of gases in this closed vessel, in this closed apparatus, and they introduced an energy source. And in this case they introduced basically like an electric spark, like what you see here. By introducing an electric spark to all of these gases in that closed environment and then cooling those gases, they realized little collections of this white precipitate, this white little collection that was developing at the bottom of the apparatus. And when they did further tests on what those little flakes were, they realized that they were actually amino acids. So they conducted this test not really having any idea what was going to happen. They basically just tried simulating the early Earth's atmosphere and then introducing a spark, introducing some energy, and just seeing what came out. What came out was actually amino acids. So this started the idea, and this was done back in the 50s. This started the idea that the first organic compounds were amino acids. And then from those amino acids you had proteins, and then from those proteins you had the development of the remaining organic compounds. And that was kind of universally agreed upon for quite a while because they were the first ones that really were able to physically prove you can make organic compounds from these inorganic components. By the way, that electricity is just there to simulate something like a lightning bolt that would have been very common in the early Earth's atmosphere because things were very erratic, a whole lot of energy in the clouds and things like that. That energy source could have triggered the production of organic compounds. It's actually been proven that you could also produce nucleic acids in the same format. And it's a much, I think, at least from my standpoint, from a lot of scientists' standpoint, it's a much more agreed upon idea that life actually began with nucleic acids like DNA and RNA, or RNA that formed into DNA. And then from those nucleic acids, you had production of things like proteins, carbohydrates, and lipids. But this experiment is still very, very important, and we value this experiment greatly, even if the general concept may not be fully agreed upon today, because it was still the first time that it was proven in a lab that organic compounds could be produced from inorganic. All right, I hope you guys got yourselves through it, enjoyed it, get a little break before we get into the next unit. Until then, take care.